Hello, my name is Sarah, and today I will be talking about glycogenosis in dogs, also known as glycogen storage disease. The reason I chose this topic was because I am very interested in veterinary medicine and have always liked learning about medical diseases in dogs. Someday I wouldn't mind doing the research, but that is another topic for another day. In this disease, there is a deficiency of an enzyme or of its activity that prevents the body from properly metabolizing glycogen and causes its buildup. In the photos on the screen, we can see stained liver cells. On the left, we have normal amounts of glycogen storage, and on the right, we see a cell with abnormally large glycogen storage. First, I want to briefly explain glycogen and its purpose before we dive in. Glycogen is simply a glucose polymer, which means it's made by stringing together multiple individual glucose molecules. Its function is to serve as the main carbohydrate store and sugar storage. It is most important for the body's homeostasis. It works to balance blood glucose and supplies energy to the muscles. The first major storage source is the liver, and it is used to balance blood glucose. Muscle may be the second site of glycogen storage, but it wins in quantity, and that is just simply because there are way more muscle cells in the body than there are liver cells. In muscle, glycogen serves as the energy source for mechanical work. Remember, a molecule of glycogen contains a bunch of individual glucose molecules strung together. They stay that way because of two types of linkages. The first type is an alpha-1,4 glycosidic bond. This joins adjacent glucose molecules. The next type is an alpha-1,6 glycosidic bond. These join polymers together and create branching. To get all of this going, we start with an enzyme that will catalyze this glycogenesis, and that enzyme is called glycogenin. It does this by attracting the first glucose with tyrosine residues, and then we're off to start our polymer. For glycogen synthesis, step 1 is taking glucose 6-phosphate from glycolysis, and we must isomerize it to make glucose 1-phosphate. This phosphorylation traps the glucose molecule inside of the cell. In step 2, from there, glucose 1-phosphate must be activated to make glycosidic bonds. UTP is added to make UDP glucose, an inorganic pyrophosphate. In this step, UDP acts as a better leaving group, while pyrophosphate drives this reaction forward. In step 3, glycogen synthase, known as the maker of glycogen, adds individual glucose units to the existing chain, and this is called elongation. Once a chain becomes about 6 units long, branching enzymes form alpha-1,6 linkages and create a more compact way to store glycogen. For degradation, the most important part is that it works by phosphorylation. Firstly, when the body is ready to use its glycogen storage, an enzyme called glycogen phosphorylase is activated and removes units from non-reducing ends by adding inorganic phosphate. This is a very critical and irreversible step. In the muscle, glucose 1-phosphate cannot be used, so we have to transform it back. The glucose 1-phosphate can then be isomerized and converted into glucose 6-phosphate to go back into glycolysis. This is using the same enzyme we did before, phosphoglucomutase. Once we do that, the glucose is released from the cell to be used by the body. As phosphorylase works to take off units, it gets stuck before coming up to a branch point, and this is usually about 4 residues away from the branch point. Once this happens, a debranching enzyme called transferase takes off the units on the branch and transfers them to the core polymer. This process leaves a glucose that is still linked via an alpha 1,6 glycosidic bond, then another debranching enzyme known as alpha 1,6 glucosidase hydrolyzes the branch glucose by breaking the alpha 1,6 glycosidic linkage that results in a free glucose to be used. Now that we have the basics out of the way, let's get into glycogenosis. Glycogenosis, as we said, is characterized by excessive accumulation of glycogen in the liver and other organs such as skeletal and cardiac muscle. This is due to defective glycolytic enzyme activity. There are 4 types of glycogen storage disease that affect dogs, but all are found in humans. In dogs, they are seen in specific breeds, and they have recently been found via clinicians and genetic testing. All of these types are caused by autosomal recessive genes. And what this means is that in order for clinical symptoms of the disease to show, a dog or puppy must have 2 copies of the abnormal gene. If they only have 1 copy, then they are only labeled as a carrier. For the first disease we have type 1A, known as von Gierke's disease. It is characterized by a deficiency of glucose 6-phosphatase that impairs the ability of glycogen degradation. This happens inside the endoplasmic reticulum, and glucose 6-phosphatase is the main regulator of glucose production in the liver. This disease was discovered by Edgar von Gierke. He was a German pathologist who specialized in glycogen synthesis. As with many toy breeds of dogs, hypoglycemia can be an issue to deal with, especially in young puppies, but this type of disease affects Maltese in much more fatal ways. The deficiency causes accumulation of glycogen in the organs of the dog. Symptoms can be emaciation from failure to get nutrients to the body, stunted growth, hepatomegaly, which means enlargement of the liver, and lethargy. Many affected dogs die in utero or at birth. This can result to stillborns and mummified puppies. If a puppy so happens to survive, it has been recorded to eventually die at about 60 days of age. Nutritional therapy has been tried, but its success only granted life for about 6 months of age. in which time they eventually succumb to severe hepatic lipidosis and pancreatitis. In a 1995 study, 2 Maltese littermates were presented for a necropsy with a history of failure to thrive, depression, and poor body condition. These puppies were found to be a small body size and emaciated with severely enlarged pale livers and pale kidneys. This was concluded to be the first confirmed report of type 1A in animals. The American Maltese Association says there are puppies that can have juvenile hypoglycemia, but that glycogen storage disease puppies will have overall a sickly appearance, whereas puppies with hypoglycemia will look and act healthy and play just like a normal puppy. They also note that glycogen storage disease puppies will have chronic low blood sugar, whereas hypoglycemic pups will actually have normal glucose levels, and only when they have a hypoglycemic attack will their blood sugar crash. Next, we have type 2, known as Pomp's disease. This is caused by deficiency of alpha glucosidase, which prevents the liver's ability to degrade glycogen and leads to its accumulation in tissues such as heart, skeletal, and smooth muscle. Researchers say it most closely resembles Pomp's disease in infants. It was first discovered by J.C. Pomp, who was a Dutch pathologist. He had first come about the disease symptoms while performing an autopsy on a young girl who died of unknown causes. The mechanism for this enzyme is hydrolyzing the terminal non-reducing ends of an alpha-1,4 linkage in the lysosomes of muscle and nerve cells. This disease occurs in Swedish and Finnish Lapphund dogs. The symptoms of this disease that closely resemble the human infantile disease are digestive difficulties resulting in vomiting, loss in muscle tone, and muscle weakness, as well as failure to thrive or gain weight, enlarged heart rhythm changes, or heart failure. In research, 4 dogs were presented with severe vomiting and muscle weakness starting at about 6 months of age. They eventually passed between 10 to 18 months of age due to cardiorespiratory failure. No Lapphund dog affected with this disease has lived past 30 months of age. In another research conducted, researchers were able to confirm a mutation in the alpha-glucosidase gene, allowing for genetic testing of other Lapphund dogs. 4 puppies from a litter were affected by slowly progressive muscle weakness and frequent vomiting. Blood samples were collected from 2 dogs, and from research done prior, the gene showing the mutation was suspected, and PCR analysis was run to confirm its diagnosis. Next, we have type 3. This is also known as Cory's disease and is due to the deficiency in a debranching enzyme known as alpha-1,6-glucosidase that leads to glycogen increase in liver and muscle. It was found by Carl and Gerty Cory, both Czech-American biochemists, who were the 1947 Nobel laureates. Later, Gilbert Forbes, an American physician, expanded on the features of this disorder even further. The mechanism for this reaction was seen earlier when we talked about glycogen degradation. This enzyme works with phosphorylase and transferase to break down glycogen polymers. More specifically, this enzyme cleaves the alpha-1,6 glycosidic bond so that phosphorylase can continue breaking off glucose from the core glycogen polymer. This disease was first known in German Shepherd dogs, but has now recently been found in curly-coated retrievers. German Shepherd dogs, as puppies, were presented with symptoms of depression, muscle weakness, hepatomegaly, and growth retardation. All 4 puppies died or were euthanized by 15 months of age. Curly-coated retrievers diagnosed and studied so far do not have such a rapid progression of disease. 2 dogs studied were presented with elevated liver enzymes in blood work at 6 and 9 months of age, and at 14 and 19 months of age started showing symptoms of exercise intolerance, weakness, and collapse. Nutritional therapy was used, and the dogs passed at 4 years of age. The last type to be found in dogs is type 7, known as Torrey's disease. It is characterized by a mutation of phosphofructokinase that impairs its ability to phosphorylate fructose 6-phosphate. This prevents the formation of ATP and results in the lack of available energy to the muscles during heavy exercise. Specifically, the PFKM subunit is deficient, impairing the ability of skeletal muscle cells to use carbohydrates for energy. It was found by Shichiro Tori, a Japanese physician, in 1965. He retired with emeritus status in 1991 from the University of Osaka and was last known as the director of Otome Hospital in Osaka, Japan. This disease has been found and studied in both the English Springer Spaniel and the American Cocker Spaniel. Springer Spaniels have been found to share similar symptoms as humans suffering from the disease, such as exercise intolerance and heart issues, but most often present with myoglobinuria and hemolytic anemia. These both cause the significant dark-colored urine. These symptoms are all seen in the classic form of the disease, first characterized in humans. In one study, an 11-year-old English Springer Spaniel was the main focus. He was one of the founding sires of a colony of PFK-deficient dogs at the University of Florida, and he had been presented with evidence of a progressive myopathy. He would not stand on his own and was unable to take a few steps without laying or falling down. This case was concluded to have symptoms resembling the late-onset form of Torrey's disease. In the Spaniel's case, it was found that his symptoms appeared to progress more rapidly than in the human type. For diagnosis of glycogen storage disease, if the clinical symptoms presented match with the suspected type of glycogen storage disease, as well as the breed it is associated with, diagnostic procedures should be continued and will vary. Tissue biopsy of organs to determine glycogen levels or blood work to evaluate liver and kidney enzymes can confirm a diagnosis. Other types of testing may include urinary analysis, genetic testing or ECG, ultrasonography, and radiography, Although diagnosis can be confirmed, the prognosis for these animals is actually poor. Nutritional therapy has been used, but it may only prolong the life of the animal for a very short period of time. This includes increased and frequent feedings of a high carbohydrate and protein diet. Genetic testing can be performed on blood or saliva from suspected dogs using PCR and genetic sequencing. The specific genes that have caused the deficiency in an enzyme have been recorded in research, and companies are able to tell you if your dog is affected, a carrier, or normal. Although this can be used for most types, Maltese testing, genetic testing, is no longer available. Breeder screening when choosing to buy a puppy is very important as well. If you are a breeder, carrier dogs should be removed from a breeding program to avoid the possibility of future cases. Thank you.